Good afternoon for folks on the East Coast, good morning for everyone west of that, and good evening if there is anyone joining us from Europe today. So today we will be discussing how to prevent cross-contamination during sample preparation. But before we jump into this, I just want to give you an idea of what this is going to look like. I'm going to keep this presentation fairly short, probably about 20 minutes. During this time, on your Zoom interface, you should have a Q&A function, and you can submit your questions. We will get those at the end. I will take a couple minutes to parse through that and answer some of the most frequently asked ones live. And then we will also collect the rest of the questions, and my team and myself will get back to you within the next week. We are recording this session, so if you like it, want to view it again, or if you want to share it with your team or other people whom you work with, feel free to ask us for the link, and we will send you, we will set you up so that you can have a copy of the recording. All right, so I'm going to present what we're going to discuss today here. So very briefly, I'll introduce the company just to get us started, and then I will talk about what I mean by sample preparation. So here, it's really having a solid sample that you need to extract microorganisms, so yeast, mold, bacteria from, and then to run bacterial analysis on this or quality control or whatnot on your sample. So we're starting pretty upstream in this microbiology process. And of course, the risk here is if you automate that process, which is something that we do at interscience, then using the same machines to handle the same samples exposes you to a risk of cross-contamination. And of course, if you have cross-contamination, then you might have false positives, i.e. results that are higher than they should be, and therefore that might force you to take preventative or corrective actions that you shouldn't have needed to take. And so that's a loss of money, a loss of time, and of course, a loss of confidence in your results. So we're going to look at step-by-step in this process how you can manage and reduce this cross-contamination risk. We'll look a little bit into what industries and what standards this applies to before wrapping it up. So interscience, for those of you who haven't heard of us, we've been around for quite a while now, just over 40 years. We designed and manufacture equipment for microbiology labs. We have a staff of just above 130 people spread over 6 different sites. So there's 2 in France. We have our headquarters, commercial headquarters close to Paris. And our R&D and manufacturing plant is in Puy-Capelle, which is in the south of France. And then going east to west here, we have offices in Shanghai, China, in Singapore, the latest one in Frankfurt, Germany, and then Boston, Massachusetts office that opened in 2002. From these sites, we export to over 100 countries. And as for myself, my name is Lauriane Sibilo. I have a background in molecular biology, and I have been the product application manager for the Boston site since 2017. So let's look at what sample preparation is. So here you see a typical process of this microbiology steps that we've sought to automate. So from left to right here, bags. That's the first thing where you're going to put your sample, and how to manage this cross-contamination is the sterile bags that you use. So we have quite a range with different sizes, with filters, non-filters, with different formats, different porosity of filters, things like that. And then gravimetric diluters to do your primary dilution, then homogenizers to extract the bacterial and microorganism content of your sample, then a whole gamut of bag tools to help you stay organized throughout this process, as well as high precision peristaltic liquid dispensing pump right here. And then on the second side of the lab, we won't be talking into details about this today, but once you've extracted that bacterial soup from your sample, you are now ready to plate with an automatic plater on your Petri dish. You incubate that, and then you count on an automatic colony counter, which is linked to a software that would analyze your bacterial load, the bacterial load of your sample. This step also lets you digitalize that process and keep a record for those of you who now comply with CFR 21 Part 11. So today we'll talk about the left part of this lab here, the sample preparation part. And the answer to this question of how do you prevent cross-contamination in this process, for me, starts with the sterile bags that you use. So there are certain traits, certain characteristics of your bags, and certain ways to use it that will let you really decrease this risk and bring it down to a level where you are fairly confident of your results. To illustrate this, I'm actually going to talk about a fairly new range that we have released that's called the bag filter, pipette, and roll. But I will walk you through all the steps that are needed so that your analysis is as precise as possible. So here are the main features of this bag that I will be referring to. So I would just want to go through them with you. The roll, when I'll talk about the sticker, the roll sticker, that's that sticker on the top. And then when you close the bag, that's a way to, a resealable way to close your bag. When you close it this way, you create a little tube, and we'll look at why that's interesting in a second. Then the second sticker here on the bottom right is the pipetting sticker. So that lets you actually open the bag and pipette directly into the bag. And then these bags have an integrated filter that let you filter out all the solid particles of your sample. So the integrated closing sticker that we looked at just now, if you close it 3 times, if you roll it 3 times upon itself, you actually get a watertight closure. Then on the right, the sticker that you peel off to access the filtrate. So this is pretty ergonomic because you can slide it in and out of a rack that we have to organize your bags. We have a video in which we present this in a second. Then the sticker to just access the filtrate for pathogens research or your quality control, your quality indicators as well. And having this integrated closure means that you can dispose of and eliminate those bags without any mess. This is all leak proof. So we have a little video that presents this particular range a little better. I will play it for you now, and right after this we will look at the different steps, step by step, how from the moment when you sample to the moment when you are ready to extract your liquid solution, I'll walk you through that process and how to make it safer. Great. So as you saw, we have 3 different models for this particular range. I'm going to get back to those towards the end, and we'll go through this. But first, let's go through the steps of this microbiological process of sample preparation. So the first step is your sample dilution. So you have your sterile bag, and now you sample directly into the sterile bag. So you put the sample on one side of the filter, right? And then you need to do your primary dilution. So these bags, these pipette and rolls, it's the 400 milliliter line. So the optimal blending volume is going to be 50 to 300 milliliters. So keep that in mind for the type of sample that you have and the dilution factor. So you slide your bag into the gravimetric diluter. And here, what you want to look out for is that there is absolutely no contact between the sample and the machine. And this is really important because you don't want to contaminate your machine with some of the bacteria that is on your first sample. And then if there happens to be contact between the machine and the next sample, then you'll have cross-contamination. So we actually have a line of gravimetric diluters where we, on top of having a machine that's made so that the sample never touches it, we have sterile tubing for each diluent. So if you were to change diluent, you could select different pumps and different tubing, so you never have to have this cross-contamination. The second step is sample homogenation. So an important thing to keep in mind here is to try and do the dilution, have a process where you can do the dilution and the homogenization in the same bag, because the fewer times you will take the sample in and out or have to transfer things, the less the risk of cross-contamination. That is really the most, and where you get the most risk for cross-contamination is when you shift things in and out of the bag. So here you're trying to extract the microorganisms from the bag. Here again, we've designed a machine so that when you put the bag in, the door actually closes it with such a pressure that you do not need to have any contact between the machine and the inside of the bag. You don't need any clip, you don't need anything to close the bag, just the pressure of the door is enough. And here, this is where it's interesting and important to have a filter, because the microorganisms will be filtered on one side of the bag, and all the solid particles will stay in the other side of the bag. Third step is enrichment, so incubation. Here, it's very important to have a watertight closing. You don't want any spill in the incubator because that could lead to cross-contamination for your next batch. Here, it's interesting to use that function that we have. So the roll bag, the roll sticker, you roll 3 times and you create a little tube. You can slide it directly on this bag rack that you have. You saw that in the video. And then you can let your incubation run and then do your pathogens research. And then the final step is pipetting. So pipetting, I know that depending on the analysis that you're going to do, if you do quality indicators research, for example, before you even do any pathogens analysis, you're going to have to open your bag 1, 2, 3, sometimes even 4 times. And every time you open the bag, you open and close it. If you have a clip, that is a risk for cross-contamination, because there's the ambient atmosphere that you're working. There's the clip. If you're working with a clip, that clip you might use from one sample to the other. It's not necessarily decontaminated. So it's really interesting here to opt for an option where you have that integrated closure. So you well, first of all, you don't have to introduce another clip that might have been used for another sample, but also the integrated closure plus the pipette sticker here just literally lets you avoid having to open it and close it. So we've made the repetitive access easy. You don't even have to open your bag. You can just pipette directly in it. And we've made it secure as well. So in other bags, usually you'll have to open from the top and pipette from the top. So you have two options here. Either you have a pipette with a really long tip, disposable, or you get pretty agile at sticking the pipette in. But there's a risk in this case that you might touch the walls of the bags and then create a risk of cross-contamination. Whereas here, we've put actually the sticker just like sitting 10 centimeters above what would be your typical, 25 gram, 1 to the 10 dilution, so 250 milliliters in the bag. And you can just pipette directly and only the cone will touch the bag, and then you discard that. And finally, the filter here is important because now we've reduced everything we could for cross-contamination. So we've reduced those false positives as much as we could. And the last thing is we don't want any particles, first of all, because they might clog the pipette. If they clog the pipette, you're not pipetting as an exact result. So your count is not going to be exact. Your concentration calculation is not going to be exact. But even if you did and you put those particles on the plate, then you create the possibility of a lot of counts of false positives. And if you filter your sample, you have much fewer of those false positives, and therefore everything that you count on the plate is what actually grew on there. So again, we're trying to keep the results, as close to the truth as we can, so that we don't end up with results that are too high and put us above the limits of our industry and require us to take steps that were not actually needed and will waste time and resources. So the next question is going to be, who is this for? Who does this apply to? And the answer, it's such an upstream process in microbiology that actually it's anyone who needs to extract microorganisms, being bacteria, yeast, and mold, from a solid sample into a liquid suspension. So that can be, food, cosmetics, environmental, but also the pharmaceutical industry and public research. So to comply with all these industries, we do also comply with multiple standards. So ISO 7218, ISO 6887, and in the North American market, the FDA BAM as well. So here are the three models that we have. You saw them in the video. So we have the bag filter roll. This one just has the sticker for the watertight closure on top. The bag filter pipette. This one just has the access to the filtrate, the easy access. And then the bag filter pipette and roll, which has both. So to conclude, let's wrap up how correctly using bags and correctly using equipment in your lab will let you manage or decrease this cross-contamination risk. So if you use the same bag for direct sampling from your production line, you eliminate the risk from having to transfer the sample from one bag to another. Then you use that same bag from dilution and homogenization. Use a bag with integrated closing so that you don't have to transfer transfer devices from one sample to another. You don't have to make sure those devices are decontaminated. And then use something where you have an easy and secure pipetting access for any repetitive analysis. Again, anytime you're going to open and close your bag, you're creating a risk for cross-contamination. So thanks for listening. I see we have a few questions that are rolling into the Q&A. I'm going to take a break in a second just to look through them. If there are questions that are coming back often, I will answer them now, so stay tuned. In the meantime, I've listed my contact here. If you want, so I want to keep those presentations more on the technical side of things. If you want more commercial information about our products, you're welcome to visit our website on interscience.com. You can get in touch with us. I'll have someone from the sales team get back to you. We have videos on YouTube about how to use our product lines, how to use them, and more videos also on those technical topics. If you are not staying with us for the Q&A, then I wish you a really nice day, and I will see you next month for another one of these webinar series. See you in a second. Okay, so I have two questions that I want to get to now because they came back quite often, and then the rest we'll get back to you after. There's also a fair amount of requests for the recording. Again, I've recorded this, so if you want a copy of this, you can message me now, or we will reach out to you or reach out to us later. And then I have some questions that are more on the confidential side, so we will reach out to you privately. A pretty interesting question is, what is the suitable incubation temperature for the labels for the stickers to work properly? So we've incubated at 37 degrees and there was no issues. If you would like to really push it and try much higher incubation temperatures, you can also reach out to us. We can send you some samples so you can see if it works for your particular application. And then another question on there is the side on which the sticker is. So that is the side that you see on the video. It's on the right side of the bag. So there's a question about, do we need to autoclave the bags? You don't need to autoclave them. They're actually sterile when we send them to you, and they're packaged in packs of 25. And so when you open the package, so just open it in a sterile environment, and you're good to go. And there's a lot of requests for the presentations, so thank you. We'll send that to you. And then a slide of the ergonomics of the bags. So let me try to get back to it, where I show how it slides on the here, this one. So at the bottom here, you see we've designed a rack. When you incubate the bags, you are supposed to leave a certain amount of space so that air can circulate between the bags and maintain homogeneous temperature. So we've designed this this bag rack slide, I believe is what we call it. And when you roll, so for the model that does not have the roll sticker, right? So the bag filter, just pipette, does not have the roll sticker. We have bag clips, and those bag clips can be slid there, or they're hollow, and they can be slid right onto those metal bars. And then for the other two models, so just the bag filter roll and the bag filter pipette and roll, so those that have the sticker, basically you create a tube when you roll it, right? So you roll it 1, 2, 3 times on itself, you close it, and you can slide this directly onto the bag rack. And at the end, you just need to slide it out and put it in the trash, and you don't need to open the bag. And again, you avoid spills both in your incubator as well as in your waste disposal. So I will close this session for now. I've noted all the other questions. We will get back to you. Thank you again for joining me today. I hope you have an excellent rest of your day or evening, depending on where you are. And we will meet again next month, and we will communicate with you what the topic of the next webinar is. Bye for now.